Hello everyone, very warm welcome to Infinity Learn NEET YouTube channel. My name is Kiran Chaudhary and I am your botany faculty at Infinity Learn. I welcome Abhishek, Virendra. Now I would love to know your name. You are typing with couples love status, so it will be really nice if you will let me know your name so that I can pronounce it better and we can have a better interaction in the class. Please let me know your name. Hello Murthy. Is there so I guess there should be some name to Murthy too. So if you're a student Murthy, please let me know what's your name. And hello Romana. Hello Saraswati. Hello Virendra. Okay, Shweta. Yeah, right, Shweta. So what's your name exactly? Couples, love status, what's your name? I'm asking your name. Please let me know. So everybody present in the class, very warm welcome to this class. Hello Yashwant. Students, as you can see, today we will be discussing about 50 must-do questions from the entire unit of plant physiology. It includes your 5 chapters. One is the first one, chapter 11th of class 11, transport in plant, mineral nutrition, photosynthesis in higher plants, respiration plants and plant growth and development. Right, my lovely kids? And then after that, we will also be discussing questions from sexual reproduction in flowering plant. So these are the chapters. We're going to talk about 6 chapters in this class, 5 from your class 11th plant physiology and 1 is the chapter 2 from your class 12. So we will be revising all the important facts and concepts on which NTA has already asked question in the history of NEET exam, right? So let us see what are those questions and let us revise those important facts and information. Remember, NTA has the history of repeating questions, so we need to revise, we need to practice at least those concepts and facts on which NTA has already asked question, because there's a high chance that they might ask it again in some other format or way in your coming NEET 2022, and that makes this session extremely important for you to attempt. That makes it extremely important for you to answer these questions, practice these questions just before your NEET, just before you sit for your NEET 2022 on 17th of July. Okay, that's fine. If you could not attend the previous class, that's perfectly fine. So that's Karishma, right, Murthy? Okay, so Karishma, on your demand, as you demanded, I have already taken classes on meiosis. And I guess you asked for a class on respiratory ETS that is already taken. Please go through the past class section, visit infinitylearn.com website, go on the past class. You will be able to access that class under grade 11th and grade 13. So change your profile if you are not, you have not entered the profile as 13 or 11. And another class that you demanded, Karishma, was on, I guess, C3 and C4 plant, and I'm going to take it tomorrow. Make it sure that you join that class, right? Hello Sunil, very warm welcome to this class. Okay, then that's fine. If you want me to call it out like that, that's okay. Okay, so anything else, my lovely kids, would you like to say? Okay, you are Yashwant. Okay, so you people keep on changing your profile name, your actual name is not matching, so it is making me a little confused. I will try my level best to remember that, but please cooperate if I mix and match. Okay, so let's start the session, my lovely kids, and also note that we have started with Infinity Learn Festival. It's a learning festival, my dear students. So in that we are offering courses at just price of $249 only. This is the lowest possible price, and you will be getting, in that course you will be getting access to live classes, mock test, doubt clarification, and also the flashcard, which will help you to revise and remember the concepts for a longer period of time. So do visit to our website and check out the amazing courses that we are offering under the Infinity Learn Festival. The offer is limited for 30 days, so please check out that. So now let's start, students. Do we have to read practical syllabus for morphology, ma'am? No, Karishma. For your NEET examination on 17th of July, just go through what all points, examples and concepts are given in your NCERT. That is more than enough. More than enough. Just stick to that. Is that okay? Karishma, is that okay, my dear? Is that okay? So let's start the class now. So as I always say, if you are new to our channel, do consider subscribing to Infinity Learn NEET channel, so that whenever in future we will go live, upload videos, give notifications, my dear students, you also get notified and you do not miss any single session in future. So as I told you, what are we going to do in this class? We will be practicing 50 must-do questions from plant physiology, that has 5 chapters in it, and the sexual reproduction in flowering plant. Let me tell you kids, this is the most important chapter in your entire botany syllabus, because if I talk about the total number of questions that are already asked by NTA in NEET exam from 2013 to 2021, if I talk about, they are roughly 40 questions, and 40 is a very big number, right? So 4 to 5 questions you can easily expect from this chapter itself. So that's the power of this chapter. Do not skip this, right? Please practice concepts, revise concepts related to sexual reproduction flowering plant. It's a very high probability that you will get definitely 3 to 4 question minimum from this, and if your day is good, you can get 4 to 5 as well, right? It's very important. So let's start now. And I will really appreciate if you will participate in the session by giving answers. So it doesn't matter whether your answer is right or wrong. What all matters is you try. If your answer is right, great. If your answer is wrong, I will help you in correcting it, and I can assure that you won't make the same mistake again in the NEET 2022. But for that to happen, first you have to try, and second you have to understand, you have to listen me carefully, what's the right solution and how to get the answer. So I hope this much is clear to everybody, guys. Chalo, let's start it. First we will take questions from plant physiology. First question, this question is from your transport, so we'll go chapter wise. First transport in plants, then chapter number 12, mineral nutrition, photosynthesis, respiration, plant growth development. It will go like that. So which of the following criteria does not apply to facilitated transport or facilitated diffusion? So these are the 4 points, these are the 4 criterions. Can you tell me which one is not applicable on facilitated diffusion? Come on everybody, everyone, give it a try. So couples, so I will pronounce your name as couples, okay? I can't say the entire one because it is going to be time consuming. I hope you're going to cooperate with me over that. Is that okay, couples? Now I'm feeling so little bit awkward in pronouncing that, but please help me in knowing your name. Why are you like, okay, give me any random name. If not, you don't want to reveal your exact name, give a random name, I'll call you with that name. Come on. Hello Syed Ahmed, very warm welcome to this class. Okay, so everybody is saying B. Let me check out. Transport saturation, yes, definitely transport saturation will take place in facilitated diffusion, because in facilitated diffusion membrane proteins are involved. So whenever membrane proteins are involved in the transport, saturation becomes an point. Transport saturation definitely occur when all the transport proteins are already blocked or busy in transporting some or the other substance. Next, uphill transport. This is in fact wrong. Uphill transport means movement of substances from their lower concentration to their higher concentration uphill. And this movement of substances from their lower to higher actually requires energy, requires ATP energy to be utilized, right? So this is not a criteria, this is not a characteristic of facilitated diffusion. This is a characteristic of active transport. So if I talk about facilitated diffusion, it is downhill. Substances move from their higher concentration to lower concentration with the help of transport protein, with the help of special membrane protein. So this one is in fact wrong and becomes answer of your question, because the right answer is it should be downhill. The right characteristic of facilitated diffusion is it should be downhill. It requires membrane proteins? Definitely. And it is highly selective because it requires membrane proteins, right? Involvement of membrane proteins makes the transport specific in nature. That's nice, great. Let's see the next question, guys. So this is, let's revise actually, so that in your exam, if any question comes from simple diffusion, facilitated diffusion or active transport, you do not make a mistake. So if you see, this is a very important table. This actually helps you to revise all the important concepts from these three types of means of short distance transport. So let's talk about, this is about simple diffusion, facilitated diffusion and active transport. Requires special membrane proteins. Special membrane proteins are required only in case of facilitated and active, not in case of simple diffusion. So that is why we say that simple diffusion is a simple diffusion. It do not require a membrane to happen, right? It can take place in a non-cellular system as well. Next, highly selective. So remember, the transport in which membrane proteins will be utilized, it will become highly selective. So since simple diffusion is not dependent on membrane proteins, in fact it is not dependent on membrane itself, that is why it is not going to be selective. But these two, facilitated and active, they are going to be selective because they require membrane protein. Transport saturation, whenever membrane proteins are involved, transport proteins are involved. Transport saturation is seen, my dear students. Since simple diffusion do not require transport proteins, so no transport saturation, but they do require, so they show transport saturation. Uphill transport means movement from a region of lower concentration to a region of higher concentration. Requirement of ATP energy will be there to happen. So because this is non-spontaneous, pushing something from the lower concentration to higher concentration is non-spontaneous. It won't happen by its own. You have to do work, right? And whenever you need to do work, that means you have to put energy, you have to it will happen at the expense of ATP energy. So uphill transport will happen in active transport, and here facilitated diffusion and simple diffusion both are downhill. Substances will move from their higher concentration to their lower concentration. Requires ATP energy only when the transport is uphill from lower to higher concentration region, then only ATP energy is required. Hence, ATP is required only in active but not in facilitated and simple. Is this much clear, my lovely kids? Everybody present in this class, is this much clear? Is this much clear? Say yes or no, give me a thumbs up. Any reaction would work, but let me know. Is this much understood? Everybody, come on. Come on guys, don't be quiet. Yes, thank you. So I think Yashwant and Shweta and Romana and Syed, right? Am I pronouncing your name correctly? Yeah. So thank you very much. So let's attempt this question now. What will be the direction of flow of water when a plant cell is placed in a hypotonic solution? When a plant cell is placed in a hypotonic solution. So let me help you out in understanding this, my dear students, and please note, hypo, hyper, all these terms are relative in nature. You can't call somebody as hypo until and unless something is hyper in the surrounding. So they are comparative words, right? So when you say that the solution is hypo, that means the cell that is placed inside a solution will be hyper because they are relative terms. So let's see this. So this is, let's say, your solution. This is your solution. Let's say this is your solution. And let's say this is your cell. Let's say this is your plant cell. This is solution and this is your plant cell inside the solution. Now we are saying that solution is hypotonic. Hypotonic. That means the plant cell also acts as a solution because, my dear students, it has water in the cytoplasm and it has solutes, especially in the vacuole of the plant cell, right? So this makes a plant cell act as a solution as well. So if this solution is hypo, that means plant cell is going to be hypertonic. Right, my dear students? When I say hypotonic solution, that means the concentration of solvent or concentration of water will be higher in the surrounding solution in comparison to the concentration of water which is there inside the cell, right? So water की concentration कहाँ ज़्यादा है? Water concentration is more in the solution surrounding the plant cell, and water concentration is lower in the plant cell. And always remember, my lovely kids, water has a tendency to move from its higher concentration region to its lower concentration. तो पानी जहाँ खुद ज़्यादा होता है से वहाँ जाता है जहाँ वो खुद कम है. Water moves from a region where it is present at a higher concentration to its lower concentration. So where water is at higher concentration? In the surrounding solution. Where it is lower? In the cell. So water will start moving in. So water will start moving inside the cell. Water will start moving inside the cell. Is this clear, my lovely kids? So water will flow into the cell. Excellent, excellent, very impressive. Everybody is on the same page. Excellent. Next, water potential of pure water is, water potential is the ability of the water molecules to move from one place to another place, from one cell to another cell. So this ability of water is the water potential, and it can be expressed, my dear students, in values as well, right? So by default, you are excellent couples. Yashwant, Shweta, Karishma, that's nice, that's amazing. What about Syed? Syed hasn't replied. Syed, you haven't replied. Yeah, you're giving me every time laughing face, huh, couples, because you're playing with me, right? You're not telling me your name and forcing me to be called to call you as couples. You people play with the teacher as well, huh? You can help me in pronouncing your name, right? But you're not doing that. Okay, I'll try to adjust. You're not cooperating then. Water potential of pure water is, my dear students, by default 0, and 0 is the maximum value of water potential. Maximum value of water potential. Okay, so always remember, whenever there is formation of a solution inside a pure water where you, or solvent, if you add solute, the water potential goes down of the pure water. So maximum value of water potential is for a pure water, and that is by default set as 0. And if you go on adding solute in it, the water potential decreases, hence it goes into negative number. So please note, water potential of all the solutions will be in negative, because the maximum value of the water potential is 0, which is for the pure water. And on addition of solute, water potential decreases. Is this clear, my lovely kids? Is this clear? Water potential of pure water is 0, and this is the maximum value. This is the maximum value of water potential by default. By default it is set. तो अब इसके नीचे तो negative में जाएगी. Is this clear? Next, the process responsible for facilitating loss of water in liquid form from the tip of the grass blades at night and in early morning is. So can you tell me because of which force, because of which factor this process takes place? And please help me know in the chat box what is the name of the process, as well as choose which of the following process is responsible for this. Come on everybody. Karishma, Syed, Yashwant, couples, Shweta, come on kids, Sunil, come on, give it a try, my lovely kids. Okay, come on. Fine, so you guys are right. You guys are right. It is root pressure. It is root pressure. So root pressure is actually responsible for guttation. Guttation is loss of water in form of water droplets. right? It is seen in night or in early morning, where rate of transpiration is quite less but rate of absorption of water is high. So there is more and more water getting accumulated inside the xylem, and this excess water get lost because there is no transpiration. Water is not getting lost in form of vapor because there is no sun, right? There is no sun, that means water from liquid phase is unable to convert into vapor and cannot escape because it is night or early morning, there is no sun over there, right? So that is why, my lovely kids, this is the most favorable time to lose water, excess water in form of droplets, and the process is guttation and it takes place due to root pressure. Excellent. Hello Virat, you people have excellent names, na? Names of your like IDs, Virat Kohli 07, impressive. Chalo, next. Xylem translocates. So this is an amazing question, and you need to remember this information. Let me see if you can crack this one. What do you think? Xylem translocates what? Just water, mineral plus water only, or there can be something else also that can also get transported from xylem? Hello Vijaya, very warm welcome to Infinity Learn. So Yashwant is saying, are you a faculty at Sri Chaitanya College, madam? I am working at Infinity Learn, my dear students, which is by Sri Chaitanya. So yes, I work for Sri Chaitanya. Vijay Krishna, okay, fine. So I guess you guys are giving the right answer. Hello Indrajit Chaurasia, very warm welcome to Infinity Learn. Yeah, so xylem translocates water, mineral salts and some organic nitrogen and hormones also. So this is something you need to remember. Generally we have this perception that if it is xylem, it will translocate water and minerals only, but that is not the case. Along with water, mainly it is water and mineral, but along with that some organic nitrogen and hormones are also transported. This is a tricky question, please remember this. Excellent, everybody is on the same page. I am feeling so good by seeing your answers to be exactly same and correct. Very good. Next question. Which of the following facilitates the opening of stomatal aperture? So stomata is opened. This opening and closing of stomata is aided by which of the following feature? Come on everybody, Mohit, Yashwant, Syed, Vijaya, Indrajit, Hansika. Okay, Hansika is also there and Vijaya Krishnan is also there. So you guys are related? Your surnames are same. Yes, couples, you're right. Everybody's right over here. Karishma, yeah. What about Shweta? Shweta, you haven't replied, my dear. Okay, everybody's on the same page. That's absolutely right. Absolutely right. So that's radial orientation of cellulose microfibril in the cell wall of the guard cell. So if you see, my dear students, this is how the guard cell look like in an open form. And this opening of the guard cell is aided, that means it is helped by the radial arrangement of the microfibrils. So when these microfibrils, they're going to contract and relax, so they're going to open and close the stomata. Next. A column of water within xylem vessel of tall trees, I repeat myself, a column of water within vessel xylem of tall trees does not break under its weight because of, so see my dear students, imagine a eucalyptus tree, how tall it is. Imagine a gymnosperm, red like a giant sequoia. So I'm just trying to help you. Imagine a coconut tree, palm tree. They are so tall, so tall, right my dear students? So what do you think? How it is possible that the water is flowing in the xylem without breaking? There's an enormous gravity working, but still the water is not breaking. So what do you think is the force responsible for this? Everybody, excellent. You guys are making me cry now. Excellent. I mean, everybody is on the same page. I'm feeling so nice. Very good students. You're going to nail this exam, NEET 2022. At least for this chapter, definitely. At least for this unit, definitely. Let me see how good you are in the coming chapters, units as well. Excellent, excellent. So, high tensile strength of water. So, let me help you in understanding, my lovely kids. This high tensile strength of water is responsible for holding the water straight against the gravity. So, because of high tensile strength of water, the water column does not break because of its weight in the xylem. Now, the point is, who is responsible, my lovely kids? Now, the point is, ये गलत हो गया, that's okay. Couples, that's perfectly fine. Now, what I told you, if your answer is right, excellent. But if your answer is wrong, you have to at least try, which you did. Now, the second thing is try to listen the correct explanation, and I assure you, you will not make the same mistake if this question or related to this comes in your NEET exam, and you do not have to delete your answers if it is wrong, right? Try to learn from your mistake. Don't hide your mistakes. Hiding mistakes will not help, right, couples? Is that okay? Yes, don't hide the mistake. If you make your mistakes open, you learn from it and you don't repeat it. So, see this, high tensile strength of water. I am teaching this, couples. Now, listen this very carefully. I will make it super simple and clear for you. High tensile strength of water. High tensile strength of water is because, now listen, this high tensile strength of water is responsible for not allowing or making the water column break. So, water column is not breaking in xylem because of high tensile strength of water. Now, I am explaining you how this high tensile strength of water is built up. Why there is high tensile strength of water in the xylem? It's because of three things. First is cohesion. Because of these three factors: cohesion, adhesion and surface tension. Now, listen this, my dear students. What is cohesion? One water molecule is getting mutually attracted to the another water molecule. तो एक water molecule और दूसरे water molecule में attraction है. So, this water molecule which is below, it is attracted by the water molecule which is above. So, in this way, the water molecule which is above in the xylem is pulling the water which is below. So, this is how they are helping the whole stream of the water to get lifted above. So, that water can move from root xylem to stem xylem and finally from stem xylem to leaf xylem. So, this whole, and against the gravity, when the water is traveling from stem xylem to leaf xylem, this against the gravity, when the movement is there, the mutual attraction that is there between the two water molecules help them to lift up against the gravity. Another mutual attraction force is between water molecules and the tertiary elements, means the elements of the xylem, right? So, the elements of the xylem, the vessels, the tracheids, they are actually showing mutual attraction with the water molecules, right, my dear students? So, when a water molecule is trying to get lift up against the gravity in order to travel upwards, my dear students, this additional force other than cohesion, this my dear students, adhesion force, that is the mutual attraction between the walls of the xylem, my dear students, and the water molecules, this also helps in lifting upwards the water molecules. So, cohesion, adhesion helping the water molecules to lift up. Cohesion is between the water molecules, adhesion is between water molecules and the walls of the xylem, right, tracheids, vessels, etc. Now, listen this, my dear students. The third thing, that is the surface tension. Surface tension, you have already studied this in your physics and your fluid mechanics, my dear students. Surface tension is more when water is there in the liquid phase in comparison to when water is there in the vapor phase, right? So, this water which is traveling in this xylem, it is in liquid phase, right? There are water molecules in the liquid phase and not in vapor phase. So, this additionally, my dear students, this gives a higher value of the surface tension, which gives higher capillarity. So, because of these, all the three, cohesion, adhesion, surface tension, my dear students, there is high capillarity. Water rises up in the thin tubes of the vessels against the gravity without breaking. Is this clear, my lovely kids? Couples, you made a mistake. I hope after this you are not going to do this, right? So is this question clear now, that these are the physical properties of water: cohesion, adhesion, surface tension, because of which water has a high tensile strength, because of which it do not allow the water column to break even against the gravity. Indrajit said, please repeat the second point. Which is the second point? Please let me know. Yashwant, okay, perfect, clear, perfect. Couples, is this clear? Indrajit, please let me know which is the point that you are pointing out. I would love to explain it again. Please be more specific in the chat box. Indrajit, fatafat batao. Indrajit, jaldi batao. Are you asking about adhesion? Indrajit, are you asking about adhesion, my dear? जल्दी बताइए. अच्छा, cohesion के बारे में. See, listen, my lovely kids. Indrajit, cohesion is the mutual attractive force between two water molecules. So, एक water molecule दूसरे water molecule को ऊपर की तरफ खींच रहा है क्योंकि उसमें attraction force है. So, right, cohesion means attractive force between two molecules of the same kind. तो water molecule same kind है, right? And also remember, दो water molecules में आप अपनी chemistry का knowledge लेकर आओ, hydrogen bonding होती है, right? So one water molecule attracted or bonded to another water molecules with the help of hydrogen bonding. So this is acting as a mutual attractive force, cohesion, which is pulling the water molecules upward. Now everybody is requesting, but now couple has actually gained attention, attraction, right? That is why now I don't even know you are he or she, but now you're not going to reveal, but this is very much clear. So leave it now. He or she is not going to reveal the name. Leave it. So we have to adjust with this term couples now. Oh my god. Okay, clear? Okay, fine, perfect. Now let's see the next question, kids. Next question. What is the direction of movement of sugars in phloem? Come on everybody. Direction of movement of sugars in phloem. Give it a try. कोशिश करेंगे सभी लोग. Come on, give it a try. Thank you, Yashwant. चलिए, give it a try, kids. Give it a try, kids. Everybody, come on, everybody. Yes, come on, kids. Yes. Okay, that's perfect. It is actually bidirectional. That's absolutely right. So remember, kids, direction of movement of substances in xylem is unidirectional from root to the leaf. But when it comes to phloem, the movement of substances, sugar molecules is or the food is actually bidirectional. Why it is bidirectional? Because inside the same phloem element, my dear students, in the sieve tubes, the food can move in both the direction. That means from source to sink, definitely the food moves, but sometimes the relationship of source and sink is actually convertible, it's reversible. In some particular season, in some particular condition, the plant part or organ which is acting as a source, in some next season it can act as a sink. So because source and sink relationship, you are absolutely correct when you think that food goes from source to sinks. That's absolutely correct. But the source and sink itself are interconvertible, they are reversible. It depends upon the season, condition required in the plant. So whenever source and sink, this relationship changes, the food can flow in the other direction as well. So right now, if this particular organ is acting as source, in some other season it can act as a sink also, so the food will backflow. So please remember, my dear students, direction of movement of sugars in phloem is bidirectional, but movement of water and minerals in phloem is unidirectional. Excellent. Now let's see this. Come on everybody, let's see this. Next question. So we are done with all the important questions from transport in plant. Now let us practice some important questions from mineral nutrition. So let me see who can get. So you, by far, you have been doing excellent in chapter number 11, transport in plant. Let me see how good you are in chapter number 12, mineral nutrition. So let me know what is the right answer to this match the column. So in column A, you have elements. In column 2, you have their roles. Come on everybody, give it a try kids. Koshish kijiye. Everybody, come on kids, everybody, give it a try. Come on kids, everybody, give it a try. So Pushpa is also there on our platform. Hello Pushpa, very warm welcome to Infinity Learn. Okay, let me see then. You guys are giving answer C. Most of you are giving the answer C. So let me see if the answer is C or not. Okay. Iron. Iron is required for the synthesis of chlorophyll, that's right. Zinc is required for the biosynthesis of IAA, which is auxin. And boron is required for pollen germination. Manganese is very much required, very important role it has in photolysis of water, which is going to release oxygen, which is one of the byproduct of photosynthesis. Yashwant. Okay, don't say that Yashwant. Come on, let us be more serious on these questions. Iron. So iron A goes with 3. Yes, B goes with, zinc goes with 4. And C, boron goes with 2 and manganese goes with 1. That's absolutely correct. Excellent everyone. Next question. Which of the following elements is responsible for maintaining the turgor in the cell? Turgor in the cell means incoming of the water. So which of the following element is responsible for endosmosis and exosmosis? That means maintaining the turgidity of the water molecule. Whose concentration, change in concentration is going to affect the incoming and outgoing of the water? Come on everybody, yes, come on, come on. Are you sure it should be sodium? You everybody is giving sodium, 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 sodium. A, B, C, D, A, B, C, D भी सही लिखा है. Are you sure it should be sodium? बेटा हम neurons की बात नहीं कर रहे हैं. We're talking about plants. We're talking about plants. We're talking about plants, not neurons. आप लोग शायद zoology में चले गए. Yes, that's absolutely correct. Now you people have come back to botany, I guess, from zoology. Yeah. So it is potassium, K. Right. So potassium, change in potassium ion concentration is going to change the turgidity. It is going to, it will be responsible for inflow of the water into the guard cell and also removal. So opening and closing में भी ये help करेगा. Right. Potassium theory, you remember? Oh, that's fine. You don't have to be sorry, my dear students. I just expect you to learn. If you make a mistake, that's it. Hello, Akula, Kavita. Hello, Kavita. Very warm welcome to Infinity Learn, my dear. Next question. Deficiency symptoms of nitrogen and potassium are visible first in. Deficiency symptoms of nitrogen and potassium are first visible in. Come on, everybody. Give it a try. देखते हैं कि इसका आंसर आप सही दे पाते हैं या नहीं. Come on, it's a logical question. Yes, kids, come on, everybody. Indrajit, Kapil, Syed, Kavita, Pushpa, Karishma, Yashwant, Shweta. Come on, everybody. Give it a try. Hansika. Good. That's absolutely correct, my lovely kids. Very impressive. Hello, Ramvant, Dattatray. Very warm welcome to Infinity Learn, my dear kids. Very warm welcome. That's right. Everybody is on the same page. Thank you. That's right, Virendra. It is senescent leaves. So what happens? Nitrogen and potassium, my kids, they are highly mobile elements. They are highly mobile elements. So whenever the elements are mobile in nature, whenever a plant part is about to die or undergoing senescence, undergoing agents, about to fall. In that case, the mobile elements are withdrawal from such plant parts because they are going to end up dying. That is why they are removed from them, they are withdrawn from them, and they are given or redirected to the plant parts which are growing like buds, like leaves which are in developmental stages, right? This is how plants and cells utilizes the available resources smartly, right? So such mobile elements deficiency will be seen in plant parts which are showing aging or senescence, about to die. Excellent. Yeah, that's Shweta. Thank you, Shweta. Next, the products of reactions catalyzed by nitrogenase in root nodule of leguminous plant is. What are the products of biological nitrogen fixation? What are the products of biological nitrogen fixation? That's a very good question and conceptual. Let me see if you can crack this. It's a little tricky, huh? First only I'm telling you. It's a little tricky, guys. It's a little tricky. Can you get this? Yeah, hello Prabha. Very warm welcome to Infinity Learn. Do not hide your mistakes. Accept the mistakes and learn it and don't repeat it. When you will hide it, you will repeat it. If you don't hide it, you will not repeat it. Learn this from now, right? That's absolutely correct. It should be ammonia and hydrogen. So not only ammonia during biological nitrogen fixation, nitrogen which is present in the atmosphere in N2 form it is converted into ammonia, right? But during this biological nitrogen fixation, not only ammonia but hydrogen is also produced. Please remember this. That's amazing. So this is the reaction that take place which is catalyzed by the enzyme nitrogenase which is present into the rhizobium, right? In that case it was biological nitrogen fixation in your NCERT is explained with the help of leguminous plant, and the organism which is helping in or which possess this nitrogenase enzyme is the rhizobium bacteria, which is growing inside the roots by forming nodules in the leguminous plants. So in this, my dear students, this is your atmospheric nitrogen. It is converted into ammonia. So one N2 is converted into 2 molecules of ammonia, but along with that hydrogen is also produced. Is this clear? My name is Kiran Chaudhary, Indrajit. My name is Kiran Chaudhary and I am your botany faculty at Infinity Learn. I love teaching botany. Okay, my name is Kiran Chaudhary. I would like to correct the spelling that you have written. It is Kiran, K-I-R-A-N-C-H-O-U-D-H-A-R-Y. Is this clear? Okay, now let's get back to the question. One very important thing. Please note, my dear students, one very important thing, that is ammonia is the first stable product during biological nitrogen fixation. This is also given in your NCERT and NTA has asked this question in the NEET exam as well. So the first stable product in biological nitrogen fixation is ammonia. So ammonia is the first stable product. Right, kids? Is this clear? In biological nitrogen fixation. Come on. Okay, so somebody is new at our platform. Dharani. Hello, Dharani. Very warm welcome to our platform, Infinity Learn. Let's practice some more questions, Dharani, and I will really appreciate if you will participate in the session by trying to give the answers to this question. So let's do this, kids. Thio Bacillus. Thio Bacillus is a type of an organism, a bacteria that you must have heard about in the nitrogen cycle. I'm talking about the entire nitrogen cycle. If you recall, you'll be able to recollect that this bacteria you saw somewhere in the nitrogen cycle. So can you help me in which process Thio Bacillus bacteria helps during nitrogen cycle? in the nitrogen cycle? Come on everybody, you people are absolutely correct over this. Thiobacillus helps in denitrification. What is denitrification, my dear students? Denitrification means conversion of nitrates. The nitrates NO3 minus which are present in the soil, right? So, my dear students, although while doing nitrogen fixation, the atmospheric nitrogen is somehow converted finally into, my dear students, the nitrates, which is the most absorbable form of nitrogen by the roots of the plant. So, the basic aim is to convert it into the nitrate, right, so that plants can easily absorb from the soil. But a very small amount of nitrate, which is actually produced via fixing in the soil, my dear students, which is added into the soil by nitrogen fixation, a little amount of it is back converted into nitrogen. It is back converted into nitrogen and released into the air. But this will take place at a very minute amount. Otherwise, the whole process or doing the exercise of converting atmospheric N2 into nitrate will become futile, waste. It will all go in vain, but that doesn't happen. Hello Ganesh, very warm welcome. Ganesh is saying I found you on Instagram. Thank you very much Ganesh, and I will really appreciate if you will participate in the session as well by trying to answer these questions. Hello Savita, बहुत late हो गया बेटा, but whatever you missed out Savita, it's my kind request, please go and check out that. That's absolutely right Prabha. Pseudomonas and Thiobacillus are the two bacteria that helps in this denitrification, conversion of the nitrates back into the nitrogen N2 and releasing into air, but this will happen at a very low scale. Okay, so this is catalyzed by, or this is done by bacteria like Thiobacillus and Pseudomonas. So these are two important bacteria given in your NCERT, going to do denitrification. Clear guys? Clear my lovely kids? Chalo, let's see the next question then kids. During biological nitrogen fixation, inactivation of nitrogenase enzyme, this is your molybdenum iron protein, this nitrogenase enzyme, which is the most important and essential enzyme to convert N2 into ammonia. So, inactivation of nitrogenase by oxygen poisoning is prevented. So, this nitrogenase enzyme is quite sensitive to molecular oxygen O2. So, which is the following substance which helps in maintaining the anaerobic condition in the place where the nitrogen fixation is taking place? Who acts as an oxygen scavenger there? Come on everybody, give it a try kids. Come on everybody, give it a try. That's absolutely correct. It is leg hemoglobin. This is the scavenger. It eats up whatever oxygen is available in the vicinity of the nitrogenase, so that nitrogenase activity is not stopped. Scavenger of molecular oxygen. Next. Yeah, so we are done with all the important questions, my lovely kids, from mineral nutrition. Now we will practice some very important. So I want okay, these were the important questions. That's okay. Couples, you can see this recording in 2x speed. But if I'm going to give you the practice in 2x speed, I doubt what you will be able to absorb from this session, right? So now we are done with the questions from mineral nutrition. We will be doing questions now from photosynthesis. This is a very important and conceptual question. Let me see who can crack this one. So one scientist cultured cladophora in a suspension of azotobacter, which is a aerobic bacteria, and illuminated the cultured by splitting light through a prism. He observed that bacteria accumulated mainly in the region of. So can you find out which one is the right answer kids? Come on everybody. Yes, come on everybody, give it a try kids, give it a try. Yeah, so what happened is, through using a prism, so why prism is used? So when prism is used, my dear students, the light which appears to be white in color, it breaks into its components. It breaks into its component. So visible light breaks into its component of Vibgyor, violet, indigo, blue, green, yellow, orange, red, like that. And when this Vibgyor or this colorful light actually falls, my dear students, on the filament of Cladophora, so what happens? Lot of bacteria gets accumulated in two regions. Lot of bacteria get accumulated in two regions. This is the algae Cladophora, right? And this is the lights, different lights, my dear students. Vibgyor, right? Violet, indigo, blue, green, yellow, right? Orange and red, right? So he saw that the regions of the algal filament were blue and red lights were falling, lots and lots of aerobic bacteria, azotobacter, got accumulated. So what does this signify? They were on the other sides as well, but at maximum they were accumulated where red and blue light was falling. The reason is, my dear students, these are the two lights at which rate of photosynthesis is maximum. If rate of photosynthesis will be maximum, that means production of photosynthetic products will be maximum. So who are the products of photosynthesis? Carbohydrate as well as oxygen. And these are the aerobic bacteria. They require oxygen in their cellular respiration process, in the metabolism. So please remember, my dear students, since they require oxygen, so they will accumulate, they will grow, they will multiply in that region more, where more and more oxygen is getting release. So it will take place where photosynthetic rate is higher. So with this, my dear students, the action spectrum, the first ever action spectrum could be understood, could be concluded, was given, my dear students. So please remember, the role of azotobacter was to find out where exactly the maximum oxygen is getting evolved. That will indirectly, my dear students, or directly indirectly, is going to affect or is going to show the rate of photosynthesis. Is this clear, everybody? Is this explanation satisfactory for everyone? Is this explanation satisfactory for everyone? Come on, everybody, is there any doubt in this? Okay, so I did not give the answer. Blue and red. Clear? Is this clear? Okay, perfect. Now let's see the next question then. Oxygen is not produced during photosynthesis. Again, this is a very beautiful question. Let me see if you can crack this one, and let me see if you really know the concept behind it or not. If not, then please try to understand with full patience when I'm going to explain this. Come on, try this question, everybody. Hansika, Syed, Shweta, Karishma, Savita, Couples, Syed. Come on, everybody. Yashwant, Hansika, try to give the answer, kids. Come on, try to give the answer. Oxygen is not produced during photosynthesis by. You people are absolutely correct. Everybody is absolutely correct. Excellent. They are green sulfur bacteria. So what happens, my dear students, whenever instead of water, H2S is the hydrogen donor. Whenever H2S is the hydrogen donor, instead of water, what will happen? H2S will break, it will give out its hydrogen, right? So whenever H2S is used in place of water as a hydrogen donor, so when this H2S undergoes oxidation, it produces either sulfur or compounds of sulfur, my dear students, depending upon the type of organism in which this photosynthesis is taking place. But if there would have been water as the hydrogen donor, so when water get oxidized, it will release oxygen. But if H2S is the hydrogen donor, it will not release oxygen, it will release sulfur or any other compound of sulfur depending on the organism. So in green sulfur bacteria, purple sulfur bacteria, oxygen is not revolved because water is not used as a hydrogen donor. Instead, H2S is used, which is going to undergo oxidation. Clear, my lovely kids? Tell me, is the concept clear? Everybody present in the class, is this concept clear? That's very much important for me. more than the answer. Is the concept clear? If the concept is clear, please let me know in the chat box. Yes, come on. Everybody, let me know in the chat box. Is the concept clear? Let me know in the chat box. Is the concept clear? Let me know in the chat box. Is the concept clear? Yes? Excellent. Now let's see the next question. Which of the following is not a product of light reaction or photosynthesis? Which of the following is not a product of light reaction in photosynthesis? Which of the following is not a product of light reaction in photosynthesis? Come on everybody, give it a try. I'll have a sip of water. You can try this question. This is a little tricky question. This is a little tricky question. Let me see who can crack this one. Hansika, you need to see this question again. Product of light reaction. Just see this one again. Yashwant, Hansika, can you see this question again? Savita, please see this question again. Not a product of light reaction. We are not talking about cyclic, non-cyclic. We're just talking about light reaction in general. Okay, so let's see this. Kids, which of the following is not a product of light reaction? So if you see ATP, NADPH and oxygen, all these three are products of light reaction. They all are produced during non-cyclic photophosphorylation or photosynthesis. But NADH, this is produced during cellular respiration, right? Not photosynthesis. Is this clear, everyone? Don't make this mistake again, okay? Is this clear? Hello, latest gamer on Infinity Learn platform. May I know your name, latest gamer? Next question. Phosphoenol pyruvate PEP is the primary CO2 acceptor in. Phosphoenol pyruvate PEP is the primary CO2 acceptor in. Come on everybody, give it a try. Come on everybody, give it a try. Phosphoenol pyruvate, it is a 3-carbon compound. It's a 3-carbon compound. It's a 3-carbon compound. So phosphoenol pyruvate is a primary CO2 acceptor in which type of plant? Everybody, hello Krishna Suri, very warm welcome to Infinity Learn. Please try this question everyone. Come on. Phosphoenol pyruvate PEP is the primary CO2 acceptor in. Is a primary CO2 acceptor in. That's absolutely correct. It should be in C4 plants. It should be in C4 plants. Krishna, we're talking about PP, PEP, 3-carbon compound. It happens in C4 plant. It happens in C4 plants. Let me help you in understanding this. In C3 plants, Krishna and other students who thought the answer is D, in C3 plants, just the C3 cycle runs where Rubisco enzyme makes CO2 react with RUBP, which is a 5-carbon compound. Right? So the first CO2 acceptor, primary CO2 acceptor in C3 plant is RUBP, a 5-carbon compound. Right? Okay. When it is the case of C4 plants, there are two plants that participate in photosynthesis, mesophyll cell and bundle sheath cell. In mesophyll cell of C4 plant, primary carboxylation takes place and the first CO2 acceptor is PEP, 3 carbon compound. Second time the fixation takes place inside the bundle sheath cell where the CO2 is accepted by RUBP again, but that is the second CO2 acceptor, not the first one in C4 plant. Is the concept clear, Krishna? No, do not relate your answers, my dear students. I told you 100 times, accept the mistakes. When you accept it, you won't repeat it again. Clear, kids? Clear, everybody? Is the concept clear? Please let me know in the chat box. Is the concept clear? Yes? So, in the mesophyll cell of C4 plants, just the light reaction will take place. The dark reaction will take place. Rubisco is present in the bundle sheath cell. Rubisco is not there in the mesophyll cell of C4 plants. Understood? That's perfectly correct. Now let's see the next question, kids. Then, a plant in your garden avoids photorespiratory losses. Now the question is finished here only. Thank you very much, Yashwant. Thank you, Shweta. So thank you, Krishna, Savita, everybody. So what you need to understand over here is the question itself finishes at this part. This avoids photorespiratory losses. So it's the only one type of plant in this, my dear students, that can avoid photorespiratory losses. That is the C4 plant. That is the C4 plant. But you need to understand, but there are few, you might get confused. Okay, this is not C1, this is C3 actually. But you might get confused between other options like chem, right? Okay, this first one might help you in giving the logic that between C3 and C4 it should be C4. So you should read out the remaining statements to confirm it should be a C4 and do not get confused with the chem plants. A plant in your garden avoids photorespiratory losses. It has improved water usage efficiency, show higher rates of photosynthesis at high temperature and improved efficiency of nitrogen utilization. Whichever one of the following is the physiological group, it should be C4. So rest all information helps you to choose better and go for the C4 plants. Next question. Bundle sheath cell, as I was saying, bundle sheath cells that are present in C4 plants, what will they do, what will they possess? Let me tell you, the dark reaction will take place inside. The formation of glucose, formation of food will take place in bundle sheath cell. Now try to give the answer to these questions. Come on, everybody. Come on, everybody. Come on, everybody. Give it a try. What do you think is the right answer? Come on, what do you think is the right answer, everyone? Give it a try. Where is the Josh? I think Josh आपका नीचे चला गया है. Don't make me shout to raise your Josh. Come on, everyone. कोशिश कीजिए. Try to give the answer. Hello, Arvind, Shivran. Very warm welcome to Infinity Learn. So what do you think is the right answer, Arvind? Arvind thinks they are rich in Rubisco. Okay, Arvind, Krishna, Savita, everybody, Shweta, excellent. The answer is this, excellent. They will be rich in Rubisco because in bundle sheath cell the dark reaction will take place, and for dark reaction to take place CO2 must react to RUBP, and this reaction will be catalyzed by Rubisco. They are rich in Rubisco. Excellent. Now see the next question. Oxygenation activity of Rubisco. So remember, my dear students, Rubisco has a dual nature. Rubisco can make, it's an enzyme, it can make oxygen as well as CO2 react with RUBP. Alright. Now it depends upon who is present in what concentration, or so it depends upon the partial pressure of the relative gases, respective gases, right? If the partial pressure of CO2 is higher in the vicinity of Rubisco, Rubisco will make O2 react with the RUBP, but if the partial pressure of CO2 is higher in the vicinity of the Rubisco, Rubisco will make CO2 react with the RUBP. But if the partial pressures are almost comparable, then it has more, Rubisco has more affinity for CO2. It will make CO2 react with the RUBP. Right, my lovely kids? So, oxygenation activity of Rubisco means photorespiration, which is a characteristic of C3 plant. So, during photorespiration, what happens, my dear students, with RUBP, who reacts with RUBP, my dear students, who reacts? Oxygen react. RUBP is the 5 carbon compound. So, as a result, my dear students, there will be formation of 2 compound. One will be made up of 3 carbon compound. One will be made up of 3 carbon compound. ओके सो एक जो आपका कंपाउंड होगा वो 3 कार्बन का होगा, दैट इज PGA, phosphoglyceric acid, right? And another will be made up of 2 carbon, which is phosphoglycolate, which is phosphoglycolate, right, my dear students? Phosphoglycolate. So this is what happens during photorespiration. This is what happens during photorespiration. Okay, Yashwant, thank you. Okay, hello Arvind. Let's do the next question then. So if you see in this, so RUBP is a 5 carbon compound, so it can produce, the products can have at max 5 carbon only, 3 in PGA and 2 in phosphoglycolate. So whenever RUBP reacts with CO2, it makes 2 compounds of PGA, but this time in photorespiration RUBP reacts with oxygen, one molecule is 3 PGA, another is of 2 carbon phosphoglycolate. So it will produce only one molecule of 3 carbon and the another one will be of 2 carbon, that is phosphoglycolate. Is this clear, my dear students? Is this question clear, everyone? Karishma, Arvind, Savita, Shweta, Yashwant, Krishna, is this question clear, guys? Any doubts in this? Is this question clear, guys? Any doubts in this? Any doubts in this? Any doubts in this, kids? Next question. Which of the following process in which, let me tell you, all the important questions from photosynthesis are over. Now we will be practicing some very important questions from chapter number 14, respiration in plants. Let's see this. Okay. So which of the following process, in which of the following process CO2 is not released? Come on everybody, give it a try. In which of the following processes CO2 is not released? Come on everyone, give it a try. Give it a try, कोशिश कीजिए. क्या लगता है आपको, क्या आंसर होना चाहिए इस क्वेश्चन का? बताइए everyone. Yes. That's absolutely correct. That is absolutely correct, kids. Which of the following process CO2 is not released? In lactate fermentation it will not, because what happens in lactate fermentation is glycolysis hoga. Glycolysis does not depend upon presence of oxygen, right? So glycolysis hoga. In glycolysis, my dear students, one glucose molecule से आपको क्या मिलेगा? From one glucose molecules there will be 2 pyruvic acid molecules, 2 pyruvic acid के molecules बनेंगे. Now these 2 pyruvic acid molecules, if I talk about, my dear students, each pyruvic acid is made up of 3 carbon, right? Now this pyruvic acid molecules get converted into lactic acid. get converted into lactic acid, and lactic acid itself is a 3-carbon compound. जब one pyruvic acid which is of 3 carbon converts into lactic acid which is also of 3 carbon, that means 2 pyruvic acid will convert into 2 lactic acid, from where the extra CO2 will be released. There is no extra CO2. So this happens in lactate fermentation. Lactate fermentation. But if you see in alcoholic fermentation, alcohol, ethanol, it is made up of 2 carbons. So one 3-carbon compound, pyruvic acid, converts to 2-carbon compound. That is why one carbon is released in form of CO2. Aerobic respiration, we all know all the carbons present in the glucose, all 6 carbon will come out as 6 CO2. Whether it is in plants and animal, doesn't matter. It will happen in the same 6 CO2 आएंगे. Alcoholic I already explained, and this is the lactate in which CO2 is not released. Excellent, everybody. Impressive. Let's see the next question then. The number of substrate level phosphorylation in one turn of citric acid cycle. What is substrate level phosphorylation? Substrate level phosphorylation means ATP is getting synthesized without chemiosmosis. That means without building the proton gradient. So ATP is synthesized when one particular substrate or one particular compound changes into another compound during a chemical reaction. There is a release of energy, and that released energy is enough to make a bond between ADP and inorganic phosphate. So in this process, in this way, when ATP is produced, it is substrate level phosphorylation. So how many times the substrate level phosphorylation takes place inside one turn of citric acid cycle or Krebs cycle? Come on everybody, give it a try. Substrate level phosphorylation, ATP भी हो सकता है और GTP भी हो सकता है. I am sure you are getting confused between ATP and GTP now. श्वेता, ATP, GTP दोनों हो सकते हैं. अब बताओ आंसर क्या होना चाहिए? Come on, where are you? Don't be silent. Arvind, Yashwant, Syed, couples, I haven't seen a reply from you. Where are you? Come on, Karishma. Karishma thinks it should. Karishma one turn. Karishma एक turn की बात कर रहे हैं. Answer change मत करो बेटा. Yashwant Singh Ma'am, see. Okay. So see my dear students, during citric acid cycle, during Krebs cycle, when succinyl-CoA converts into succinic acid, when there is a conversion of succinyl-CoA into succinic acid, there is a formation of one GTP molecule. Right? So one time substrate level phosphorylation takes place. So whether it is ATP or GTP, my dear students, you will consider as substrate level phosphorylation. Let me tell you how. Because in a coupled reaction, my dear students, GTP will back convert into ADP, and my dear students, simultaneously there will be synthesis of ATP molecule during the substrate level phosphorylation in the Krebs cycle when succinyl-CoA converts into succinic acid. Is this clear? So one time, in one cycle, one time. Clear? Clear? Clear or not? Yes or not? Say, everybody, is this concept clear? Please let me know in the chat box, is this much clear or not? Everybody in the class. Next question. Where is the respiratory electron transport system ETS located in plant? Where is respiratory electron transport system ETS is located in plants? Where is respiratory electron transport system ETS is located in plants? Come on everyone. Hello Manish, very warm welcome to Infinity Learn. Come on, what do you think is the right answer? Respiratory electron transport system is located in inner mitochondrial membrane. Excellent. The complexes, the electron carriers that will participate in transporting in the ETS, they are present in the inner mitochondrial membrane. Complex 1, complex 2, complex 3, complex 4, which is going to do the electron transfer. Excellent. Let's see next question. Which of the following statement is incorrect? That's a good question. It will help you in revising many concepts related to the respiration in plants chapter. Come on everybody. Yashwant, is the doubt clear? Yashwant, is it clear? Please let me know, my dear, in the chat box. Is this clear? Come on everybody, is this clear? Is this clear, everyone? What do you think is the right answer? Come on, Yashwant, Shweta, Savita, Karishma, Manish. Karishma, what do you think is the right answer? What do you think is the right answer? Which of those following statement is incorrect? Come on, answer do Manish, answer do. Koshish karo at least. So yes, Karishma is absolutely right over here. Yeah, that's right, Karishma. Enzymes of TCA cycle are present in mitochondrial matrix, that's right. Glycolysis occurs in cytosol or cytoplasm, right. Glycolysis operates as long as it is supplied with NAD, that is right, right. That can pick up the hydrogen atoms. It actually correlates with the Le-Chatelier principle you must have studied in chemistry, my dear students, in your chemical equilibrium, that when there are reactants available, only then products can be formed. So NAD is positive is the reactant, right? Until and unless there are sufficient NAD positive present, NADH molecules cannot be produced, the glycolysis cannot proceed. Oxidative phosphorylation takes place in outer mitochondrial membrane. That's wrong. Oxidative phosphorylation takes place. Oxidative phosphorylation means formation of ATP with the help of chemiosmosis, with the help of breaking the proton gradient that was built up by oxidizing and reducing the compounds, my dear students. NADH plus H plus FADH2 molecules oxidizes, helps in building the proton gradient, right? And that's proton gradient, in forming this proton gradient in between the two membranes of the mitochondria. This building of proton gradient consumes a lot of energy, and when this proton gradient is to break, it is going to release the energy, and the released energy will be utilized in bonding ADP with inorganic phosphate to produce ATP. So this takes place in or on inner mitochondrial membrane, not the outer one. Is this clear, everybody? Is the concept clear? Anyone? Is this concept clear, everybody? Is this concept clear, everybody? Is this concept clear, everybody? Any doubts till now, my lovely kids? Any doubts? Come on, come on, come on. Any doubts? Repeat it once again. Okay, so Yashwant, this question is about oxidative phosphorylation. Phosphorylation क्या होता है यशवंत? Phosphorylation means ADP का inorganic phosphate के साथ में बाउंड के बॉन्ड बना के ATP को बनाना. Now, whenever this ATP formation is taking place, my dear students, with the help of, it is linked with the formation of proton gradient. You remember chemiosmotic hypothesis, chemiosmotic theory, right? Peter Mitchell ने जो दी थी in photosynthesis, which is applicable on photosynthesis as well as respiration. So it says for the formation of ATP molecules, there should be production of a proton gradient, right? So proton gradient बना के हम क्या करते हैं? एनर्जी स्टोर करते हैं, और जब वो प्रोटॉन ग्रेडियंट ब्रेक हो जाता है तो उससे जो रिलीज एनर्जी होती है उसकी मदद से हम लोग क्या करते हैं? ADP और इनऑर्गेनिक फॉस्फेट के बीच में बॉन्ड बनाते हैं, ATP बना लेते हैं. जो फोटोफॉस्फोराइलेशन होता है तो यही प्रोटॉन ग्रेडियंट जो है वो सनलाइट एनर्जी की मदद से बनता है. और व्हेनेवर इट इज सेल्युलर रेस्पिरेशन, व्हेनेवर NADH plus H plus and FADH2 molecules, they get oxidized on complex number 1, complex number 2, and when the electrons runs, my dear students, at that point of time, lot of H plus ions get pumped into the proton gradient, pumped into the intermembrane space, between the two membrane space of the mitochondria. So this helps in building up proton gradient. So whenever this proton gradient is built up by oxidizing NADH plus H plus and FADH2 molecules, my dear students, whenever oxidation reduction reactions are involved in building a proton gradient, this process is known as oxidative phosphorylation. With the help of oxidation reduction reaction, proton gradient will be built. Now when this proton gradient is going to break, H plus ions will get released out. So who is going to help in coming the H plus ions back from the intermembrane space to the matrix of mitochondria? Who is going to help? CF0 particle. which is the part of complex number 5th, which is present in the inner membrane, inner mitochondrial membrane. So the CF0 particle will act as a transmembrane channel. It helps in facilitated diffusion of H+ ions from intermembrane space to matrix back. So when this protons come back from intermembrane space to matrix, proton gradient breaks, a lot of energy is released, and that energy is utilized in building, making, synthesizing ATP molecules. This is oxidative phosphorylation. This is happening with the help of CF0 and CF1 particles. CF1 has ATP synthase enzyme which catalyzes this reaction of ADP bounding with inorganic phosphate to form ATP. So the CF0 CF1 particle, which is a part of complex number 5th, it is present in the inner mitochondrial membrane. Oxidative phosphorylation is taking place on the inner mitochondrial membrane. Now tell me, is this question clear? Share the PDF, yes. So Mohammed, the PDF, everybody who was present in this class, the PDF of this class will be available on the Infinity Learn NEET Telegram channel. The link is in the description of this class, my dear students. Click on the link and join the Telegram channel. After this class, the whole PDF of this class, this annotated PDF you will get. Is this okay? Any doubts till now? Yeshwan, did you get your clarification? Are you satisfied now with the explanation? So now let's move to the next question then. Which statement is wrong for Krebs cycle? Which statement is wrong for Krebs cycle? Come on everybody, give it a try. Which of the following statement is wrong for Krebs cycle? Arvind, are you saying something? Arvind, please be more specific in the chat box where exactly you want my help. Okay, so please try this question. Karishma, Arvind, Yashwant, Manish, Savita, everybody, come on, try this question. Come on everybody, try this question, my lovely kids. Try this question. Come on everybody, try this question. Okay, no problem. Okay. Okay, so try this question now, everyone present in this class. Mohammed, कोशिश करो इस question का answer देने का. Try this question, everybody. Yes, Shweta says it should be C. So let me see this, everybody. Let me see this. Let me see this. So there you have to point out the wrong statement. Remember, there is one point in the cycle where FAD is reduced to FADH2. That is actually right, my dear students. That is actually right. Only one time in FADH2 molecule is synthesized in one turn of the Krebs cycle. During the conversion of succinyl-CoA to succinic acid, molecule of GTP is synthesized. This is also right. This is also right. The cycle starts with condensation of acetyl-CoA, which is a 2-carbon compound with pyruvic acid. This is wrong. The cycle, Krebs cycle, starts when acetyl-CoA reacts with oxaloacetic acid, which is a 4-carbon compound. 4 plus 2 makes a 6-carbon compound, that is citric acid. So this is wrong. This is wrong, and that is right. There are 3 points. The D is in fact right. There are 3 points in the Krebs cycle where NAD positive is reduced to NADH plus H plus, my dear students. Remember, from citric acid to alpha ketoglutarate acid, from alpha ketoglutarate acid to succinic acid, and then, my dear students, from malic acid to oxaloacetic acid. So these are the processes, my dear students, where the NADH plus H plus molecules are going to be synthesized. Any doubts till now? Any doubts? No need to be sorry. You just need to learn the mistake. You need to correct it, and do not make the same mistake in the NEET paper. That's the maximum I expect you in this class. Come on everyone, give it a try. Is this clear now, everyone? Karishma, Arvind, clear now? Shweta, clear? Shweta, Savita, clear? Savita. Shweta was right. Savita, is this clear? Next question. Okay. आगे क्यों नहीं जा रहा है? Oxidative phosphorylation, if I talk about my lovely kids, yeah, oxidative phosphorylation is, right now I have explained everybody. Now let me see how many of you can get this. Let me see how many of you can get this. Come on. Oxidative phosphorylation is, come on everybody. 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 Oxidative phosphorylation, how will you define it? Come on, Savita, Virendra, Arvind, Karishma. Come on, Shweta, come on. Yes, that's absolutely correct. That is absolutely correct. Formation of ATP by energy release from electrons removed during substrate oxidation. I have already given the detailed explanation of this. Do not delete your answers. Just try to learn from your mistake, Karishma, my dear. Do not delete your answers. Arvind, it should not be A, my dear. We have to focus on why calling phosphorylation as oxidative in nature. It's not substrate level phosphorylation we are talking about. Clear, Arvind? The same explanation that I gave you for oxidative phosphorylation in one of the previous questions, it follows. Let's see the next question, guys. Interesting question. The three boxes in the diagram represent the three major biosynthetic pathways in aerobic respiration. The arrow represents the net reactants to the product. Arrow numbered 4, 8 and 12 can be. All this can be what? So see my dear students, pathway A is your glycolysis. Pathway B is your pyruvate oxidation. Plus Krebs cycle. Pathway C is your ETS plus oxidative phosphorylation. Now if you see, glucose will enter into the glycolysis, and as a result what will be produced? As a result, my dear students, pyruvic acid will be produced and ATP molecules will be produced, and NADH plus H plus. I hope you can see this. Can anybody confirm that you can see all this? NADH plus H plus, right? So as a result, there will be production of pyruvic acid, ATP and NADH plus H plus. Pyruvic acid will enter the pyruvate oxidation, pathway B. It will undergo pyruvate oxidation. One pyruvic acid will get converted into 2 carbon compound acetyl-CoA. This acetyl-CoA will react with oxaloacetic acid and then enter into the Krebs cycle, will form citric acid cycle, and cycle continues, right? The ATP that is produced, my dear students, these are substrate level ATP molecules. Substrate level ATP molecules. They are produced from substrate level phosphorylation. Substrate level phosphorylation. Right, this ATP, fourth number. And also there will be production of NADH plus H plus molecules. 2 NADH plus H plus molecules, my dear students, will be produced when a 3 PGA is going to get converted into 1,3. When a PGL is going to get converted into 1,3 PGM, right, my dear students. So what you need to understand over here is the production of 2 NADH plus H plus molecules is these 2 molecules will enter the ETS. Terminal oxidation. So from cytoplasm they need to be transported to the mitochondria, to the inner mitochondrial matrix or the matrix of the mitochondria, where they will participate in ETS. These NADH plus H plus is going to oxidize on complex number 1, right? They will give the protons, electrons and energy. That is why they have a link with pathway C. Next, from this pathway B, what will be produced? From this pathway B in Krebs cycle, from this pathway B in the Krebs cycle, what will happen? In the Krebs cycle, substrate level phosphorylation will take place, right? When succinyl CoA get converted into succinic acid, there is formation of GTP which in a coupled reaction forms an ATP. So 8 represents to the ATP, but this is also substrate level. Substrate level phosphorylation, ATP, which is produced by substrate level phosphorylation. Now let's talk about these two compounds. So if you notice, my dear students, NADH, 7th is actually your 7th and 6th can be, so interchange भी हो सकता है, NADH plus H plus molecules हो सकते हैं, and they can be FADH2 molecules, right? So you can interchange also. So these NADH plus H plus and FADH2 molecules that are produced from this pathway B, they will enter into the pathway C. That means NADH plus H plus and FADH2 molecules that are produced, NADH plus H plus will be produced in pyruvate oxidation as well as in Krebs cycle. FADH2 is going to be produced in Krebs cycle. They will participate in the ETS. They will get oxidized on complex number 1, NADH plus H plus, and on complex number 2, the FADH2, and the released electron will travel in the ETS and finally get accepted by the oxygen to form water molecule, and the protons will be pumped into the intermembrane space. The energy that is released while breaking them, that will also be used in pushing the protons inside the intermembrane space to build up the proton gradient. So finally, from oxidative phosphorylation, from this chemiosmosis process, my dear students, there will be production of ATP when the proton gradient will break, right? So this 12 also represents to the ATP, but this ATP comes from oxidative phosphorylation, and this can represent to water. This can also represent to water, my dear students. And also remember, either CO2 bhi niklega. Here CO2 will also get released, but it will not be utilized in pathway C. I hope you can understand that. So oxidative phosphorylation hoga, right, my dear students. So your 4, your 8 and your 12 is ATP. 8 and 4 are substrate level phosphorylation wala ATP, and 12 is your oxidative phosphorylation wala ATP. So your answer should be ATP. Now tell me everybody, are you satisfied with the explanation? Is this question clear? Will you be able to answer this question even if the same question comes with some modification? Instead of 4, 8, 12, they ask you something else. Will you be able to attempt this question or not is my question. Is this question clear, guys? Everybody present in this class, come on. Let me know. Is this question clear? Is this question clear? Next question. Which of the following biomolecules is common to respiration mediated breakdown of fats, carbohydrates and proteins? Come on everybody. So whether fats, carbohydrates or proteins are used as respiratory substrate, which one is the common in all? तो शुरुआत कहीं से हो, ये कंपाउंड तो बनेगा ही बनेगा. It will be an intermediate. Come on everyone, give it a try everybody. Arvind, Virendra, Shweta, Savita, Manish, Karishma, everybody, that's absolutely correct. Acetyl CoA is the common, absolutely correct. Excellent. Next question. Manish, yaad rakhna ye. Which of the following respiratory quotient RQ for tripalmitine is? It's a very famous question. Come on everybody. RQ for tripalmitine is, it's not 0.7, it's 0.07 nahi hoga, 0.7 hoga. 0.7. 0.9 is for proteins and 1 is for carbohydrates. 0.8 तो ऑप्शन में ही नहीं है. 0.8 is not in option, my dear. Next. Name the plant growth regulator which upon spraying on sugarcane crop increases the length of the stem, thus increasing the yield of the sugarcane crop. So we are done with all the important questions from respiration. Now we are going to talk about the questions related to plant growth and regulators. That means plant growth development to chapter number 15. Come on everybody. Hello Balaji. That's absolutely correct. Excellent. The answer is going to be gibberellins. Savita, Shweta, ये आंसर gibberellins होगा, ethylene नहीं होगा. Internode elongation. It's gibberellin. Very famous question. Sugar cane का yield बढ़ा सकते हैं by spraying gibberellins on the stem. क्योंकि sugar cane का जो yield है, we get it from the sugar, right, which is there in the stem part. तो stem बढ़ा के हम लोग yield बढ़ा रहे हैं. Is this clear? Is this question clear, everybody? Let's take the next question then. Which of the following is not an inhibitory substance governing the seed dormancy? तो ऐसा कौन सा substance है which is not going to lead to seed dormancy? It is not going to make the seed undergo dormancy. Which of the following? Or in short, you can say which of the following is going to break the dormancy also? Which of the following is not an inhibitory substance governing the seed dormancy? Come on, everybody, be quick. देखो क्वेश्चन ठीक से पढ़ो. Not an inhibitory substance governing seed dormancy. Out of this four, which is not going to make the seed dormant, which is going to break the seed dormancy also, you can say. Come on, this is how NTA can play with you. When this is how we say playing with the language of the question. So please be clear, be cool, be calm, read the question nicely. Yes, that's right. It is gibberellic acid. It is not going to promote the dormancy. It's going to break it. Right? Is this clear now, everyone? Next. Fruit and leaf drop at early stages can be prevented by the application of. Fruit and leaf drop at early stages can be prevented. Fruit and leaf drop at early stages can be prevented by the application of. Come on, everybody. By the application of. Come on, that's correct. Right. Good, Karishma. It will be by applying auxins. So auxins actually prevent or inhibit, my dear students, the leaf drops, the early leaf drops they prevent the abscissions, early leaf drops in, my dear students, in the plant parts which are young. Right, but at the same time they promote abscission or breaking of the leaves and the parts of the plants which are old. So mature plant parts या organs में तो promote करेगा abscission को, but prevent करेगा abscission को for the younger parts. So this is how auxin works, right? So auxin is going to prevent the leaf and fruit drop at the early stages. Is this clear? Next question. Which of the following growth regulator is known as stress hormone? Which of the following is known as stress hormone? Come on, very, very famous question this is. Come on, everybody. I expect you to give right answer, everybody. That's absolutely correct. It should be abscisic acid. Remember, abscisic acid stress को develop नहीं करता है. Rather, it helps the plant to remove the stress. That's how in this context it is known as stress hormone. Not giving the stress but helping in removal of the stress. Abscisic acid. Please remember this. Next question. So we are done with all the important questions from plant physiology. These were the must questions that you should practice before sitting for your NEET 2022 to elevate your botany score. Now let us see what are the important questions from this chapter, Sexual Reproduction in Flowering Plant. Single chapter, 4 to 5 questions, very important. So let's practice this and then we will be done with today's session. So hold on yourself just for 5 to 10 minutes more. Okay, not 5 to 10, it will take 15 minutes at least. So hold yourself for 15 more minutes and let us practice the most important chapter questions, right? Most important question from the most important chapter, Sexual Reproduction in Flowering Plant. So are you guys right? Are you guys ready? Type ready in the chat box if you guys are ready. Come on, I want to see lots of readies in the chat box. Come on, are you guys ready? Shall we proceed? Shall we take the first question? Come on, Shweta, Kavita, Karishma, Shweta, Karishma. Come on. Savita. Hello, Shakir. Manish, yes, come on, ready? So let's take the first question then, guys. Here comes the first question. Plant parts which consists of two generations, one within the other. That's a good question. That's a conceptual question. Let me see how many of you can get that. Hello, Dharani. Please try this question, my lovely kid. Please try this question. The plant part which consists of two generations, one within the other. The plant part which consists of two generations, one within the other. The plant part which consists of two generations, one within the another. Come on, guys, give it a try. Yes? Shakir, give it a try, my kid. So let me tell you, let me give you a hint over here. Two generations means we are talking about gametophyte and sporo. So see over here, kids, the plant part which consists of two generations, one within the other, means you have to identify those structures in which one generation is sporophyte and another is gametophyte. That means sporophyte and gametophytic generation should be one in one another, right? That's what the question is saying. So let's find out pollen grains inside anther. Pollen grains are haploid. Pollen grains are produced from microspore. Microspore is produced by meiosis in microspore mother cell which was diploid. So pollen grains are haploid, hence pollen grain represents to the gametophytic generation. Gametophytic generation. And anther is a part of sporophyte. It's a diploid cells, my dear students. So anther represents to the sporophytic generation. Next, germinated pollen grain with two gametes. So this one follows one generation within the other. This is right. Now let's see germinated pollen grain with two male gametes. So pollen grain is also haploid. Male gametes are also haploid. So this entire pollen grain with two male gamete represents, this entire structure represents to only gametophytic generation. Right? Next, seed inside the fruit. Seed is also diploid, fruit is also diploid. So this entire structure represents to the sporophytic generation. Sporophytic generation. Embryo sac inside the ovule. Embryo sac is produced, my dear students, when there is three sequential mitosis in a megaspore. Megaspore is a haploid cell. It is produced via meiosis in megaspore mother cell which was a diploid cell, right? So whenever this haploid megaspore undergoes three sequential mitosis, my dear students, it leads to a production of a structure which has 7 cell and 8 nuclei in it. It is known as embryo sac. Each and every cell of embryo sac is haploid, hence embryo sac represents to the gametophytic generation. Gametophytic generation. And ovule represents to the sporophyte. Cells of the ovules are diploid. It represents to the sporophytic generation. I am talking about your integuments, nucellus, funicle, hilum, etc. Sporophytic generation. So if you see, my dear students, one generation within the another is seen in pollen grain and anther, and one generation in another is seen in embryo sac inside ovule, gametophyte inside sporophyte. So 1 and 4th is correct. So did you get this, my kids? Did you get this? Are you satisfied with this explanation? Did you got this one or not? Very good question. I want everybody to understand this question. If there is any single doubt remaining in this question, do let me know in the chat box. I would love to explain it. Come on guys, let me know if this question is clear or not. Let me know if this question is clear or not. Clear? Excellent. Let's see the next question then. Which of the following, in which of the following both autogamy and gaetogamy is prevented? Come on everybody, give it a try. Let me have a sip of water. You should try this. Which of the following, in which of the following both autogamy and gaetogamy is prevented? Autogamy means pollination taking place inside the same flower. Gaetogamy means pollination taking place between two flowers present on same plant. That's right, B papaya, that's right. शकील, it is B, it's not maize. मेज पे नहीं होगा, मेज मोनोशियस है, तो उसमें ऑटोगैमी तो प्रिवेंट हो जाएगी बट गेटोगैमी प्रिवेंट नहीं होगी. हेलो, how to pronounce your name? I'm little confused. Will you help me in pronouncing your name? It is Sheem. I'm unable to pronounce your name. Please let me know how to pronounce your name. I would love to pronounce it on our platform. Autogamy क्या होता है? Autogamy means pollination within same flower, and the flower need to be bisexual. Gaetogamy means pollination between two different flowers, present or born on same plant, right? So for gaetogamy to take place, flower तो दो different होने चाहिए पर plant same होना चाहिए. But if I talk about papaya is a dioecious. Plant. तो male papaya plant अलग होगा, female papaya plant अलग होगा. So sorry for my voice, my kids. मेल पपाया प्लांट अलग होगा, फीमेल पपाया प्लांट अलग होगा. तो मेल पपाया प्लांट पे मेल फ्लावर्स, स्टेमिनेट फ्लावर. फीमेल पपाया प्लांट पे फीमेल फ्लावर्स, पिस्टिलेट फ्लावर्स, राइट? सो in dioecious plant, autogamy as well as gaetogamy can be prevented because flowers, male and female flowers, do not grow on same plant. So whenever male and female flowers grow on the same plant, only then gaetogamy can be done. But in papaya, my dear students, they are dioecious. Male and female flowers do not grow on the same plant. Hence, gaetogamy can also be prevented. Autogamy तो होगा ही होगा because bisexual flower है ही नहीं. Male plant पे male flower, female flower plant पे female flower. Is this clear? Your name is. अच्छा, what's the meaning of this? मतलब this is a Hindi word, right? तो हिंदी में इस शब्द का मतलब क्या है? What's the meaning of your? That's very new for me. Please let me the meaning of your name. Next, so answer is papaya. You got this one? Everybody, is this question clear? Everybody, is this question clear? Next question then. So autogamy and gaetogamy both are prevented in a dioecious plant, which is papaya. So whenever a plant is monoecious, it can prevent only autogamy but not gaetogamy, because monoecious plant have male and female flower both present on the same plant, which is a condition for gaetogamy to happen. Next question. What type of pollination takes place in Velocinaria? Velocinaria is one such species of plant in which hydrophily takes place, but this is a case of epihydrophily. So in this, what happens, my dear students? Would somebody like to answer this one? Hydrophily, epihydrophily in Velocinaria. Come on, try this one now. I have already provided you the hint. Everybody, Karishma, Shame, Kavita, Savita, Shweta, Shakir. Come on, everybody. Give it a try. कोशिश कीजिए. That's absolutely correct, Karishma. Very good. Male flowers, which are actually present inside the water, they will be released. They will reach up to the water surface, and with the help of water currents, they will try to reach the female flower. The female flower will have a long pedicel, long stalk, because of which it will try to reach up to the surface of the water. So female flower, which is still attached to the female plant, which is very much required, my dear students. So it will reach to the surface of the water, and the male flower will try to reach in the vicinity, in the surrounding area of the female flower of Velocinaria, right? And if they come in contact, pollination will take place. Male flower has the pollen grains. So male flowers are carried by water current to the female flower at the surface of the water. Is this question clear? Okay, thank you very much. Synonymous for Kushal. Okay. Shame, Kushal. Thank you for enlightening me on this word. Next question. What is the most common type of embryo sac in angiosperm? What is the most common type of embryo sac in angiosperm? This is a very famous question, very important for your coming NEET 2022. Come on everybody. Yes, Rahul. Rahul, इस question का answer दो बेटा. बताओ, what is the answer to this question? I am very fine, Hiba. How are you, my dear? Very warm welcome, Rahul and Hiba on Infinity Learn platform. Give it a try. Try this question, Hiba. Try this question, Rahul. Yes, that is absolutely correct. Most common type of embryo sac in angiosperm is monosporic with 3 sequential mitotic division. I have already explained you this, how it takes place. Now listen this carefully. First of all, a megaspore mother cell. Listen this very carefully. First of all, a megaspore mother cell which is diploid in nature, it will undergo meiosis to form 4 haploid megaspores. Out of this 3, in most of the angiosperm, 3 will degenerate. Only 1 will remain functional. Now that functional haploid megaspore will undergo 3 sequential mitotic division. They will be free nuclear. First, only nucleus will devise, karyokinesis will take place, and in the end, when 3 times karyokinesis has taken place, there will be cytokinesis. So at the end of 3 karyokinesis, there will be production of 8 nuclei from the one from which the mitosis started. So after karyokinesis, there will be formation of wall around this nuclei, and there will be formation of a haploid structure, female gametophyte, that is embryo sac. There will be 7 cells into it, but there will be 8 nuclei, because the largest cell inside the embryo sac is the central cell, and it has 2 nuclei in it, that is the polar nuclei. Rest all the cell will have same one or single nuclei. Is this question clear? What is why? So why it is monosporic? Because the entire embryo sac is produced from one single haploid functional megaspore. So it is why monosporic, and that is why because of 3 sequential division. Is this question clear to everybody or not? Is this a question clear to everybody or not? Come on everyone. Yes, monosporic. Everyone. Next question. Which of the following statement is not true? Which of the following statement is not true? Come on everybody. Which of the following statement is not true? Come on everybody. Which of the following statement is not true? Which of the following statement is not true? Come on. Which of the following statement is not true? Read all these statements. Yes, give it a try. Everybody, Shweta, Rahul, Savita, Shame, Karishma, Hiba. What do you think is the right answer to this question, guys? Give it a try, give it a try, give it a try. That's absolutely right. Let me read out now. Not true, you were supposed to find out. Pollen grains of many species causes severe allergy like Parthenium, that's right. Stored pollen in a liquid nitrogen can be used in crop breeding programs, that's right. Tapetum helps in dehiscence. So my dear students, tapetum is actually nutritive in nature. So it will provide nourishment or nutrients. It will provide the nourishment. Please remember this. The remaining 3 layers of anther, that means the epidermis, endothecium and middle layer, they will have the individual function, but they collectively work in protecting the anther as well as helping in dehiscence. But for tapetum, which is the innermost, surrounds the sporogenous tissues and will surround the pollen grains as well. this is going to provide nutrition, not helping in dehiscence. Exine of pollen grain is made up of sporopollenin, which helps in fossilization of the pollen grain, which helps in protecting the pollen grains, my dear students, protecting the internal structure of pollen grain. Remember, sporopollenin is made up of those organic compounds. They are making sporopollenin so much resistant that it cannot be degraded by any known enzyme, acid or alkali. Is this clear? Is this clear? So all these statements are correct except C. Is this question clear, guys? Any doubts till now? Next question then. Filiform apertures is a characteristic feature of, come on, very simple question everybody. Filiform apertures is a characteristic feature of, filiform apertures is a characteristic feature of, come on everybody. Filiform apertures is a characteristic feature of, filiform apertures is a characteristic feature of, come on, come on. Come on, come on, yes, that's right, it's synergids. So filiform apertures are the finger-like projection in synergids, and it guides the pollen tube so that it can enter through the micropylar end, so that it can enter easily inside embryo sac through micropylar end of the ovule. Right, next question. Advantage of cleistogamy. What is the advantage of cleistogamy? So, hello Durga, very warm welcome to Infinity Learn platform. Now listen this carefully. Cleistogamy is one of a type of contrivance or one of the type of adaptations that promote self-pollination in plant. So there are some plants like Commelina, Viola, Oxalis. इनमें क्या होता है? These plants produces two types of flowers, Chasmogamous and Cleistogamous. Chasmogamous are the normal flowers जो कि bud stage में रहेंगे, then they will open and their stamens and pistils will be exposed in the air. Cleistogamous flowers are bisexual flowers which never ever open at all. So in that case, when a flower, a bisexual flower will never ever open, that means 100% autogamy, 100% self-pollination will take place in them, and they do not require any pollinating agents for doing the transfer of pollen from anther to the stigma. So they do not depend upon pollinators. They give assured seed setting. So इनमें seed पक्का, they give assurance that the seed इनमें बनेगा ही बनेगा, because pollination will take place, because there is no dependence on pollinating agents. 100% autogamy. Is this question clear, everyone? Next question. Pollen grains can be stored for several years in liquid nitrogen having a temperature. Pollen grains can be stored in liquid nitrogen for several years. Can you tell me at what temperature it can be stored? Everybody, come on. Just few more questions. Do not lose the momentum. Just few more questions and we will be done. Come on, don't lose that josh. Keep the josh high, my lovely kids. Come on. Sif, thode questions or few more questions and then we will be done with this. That's a very important chapter for your coming NEET 2022. That's absolutely correct. Minus 196 degrees centigrade. Next question. Attractants and rewards are required for. Attractants and rewards are required for which type of pollination? Attractants and rewards are required for which type of pollination? Come on, Hiba, Karishma, Savita, Shweta. Come on, everybody. Shame, Durga, Shakir. Come on, give it a try. Come on, don't lose the momentum. So this is entomophily. That means where insects are acting as pollinating agents. Insects are acting as pollinating agents, right? So please remember, my dear students, whenever living organisms are going to act or going to work as a pollinating agents, then these types of attractants and rewards are required. Attractants like large petals, colorful petals, right? Fragrance, right? Rewards like pollen grains and nectar. All this is required when the pollinating agent is living, when the pollinating agent can sense them. They have consciousness, they can sense these stimuli. But if the pollinating agent is non-living, like water, like wind, right? So in that case, my dear students, Non-living objects cannot sense the stimuli, they do not require all these things, right? So हवा को या पानी को, wind या water cannot sense color, smell, they have nothing to do with the pollen grains or nectar, etc. And cleistogamy does not require, because it never opens, they are not dependent on pollinating agents, right kids? Is this clear? Why the answer is entomophily? Is this question clear? Hello Sumit, very warm welcome to Infinity Learn platform, my dear. I will really appreciate if you will participate in the session by giving answers too. There are just few more questions left. Come on, let's do it togetherly. Flowers which have single ovule in the ovary and are packed into an inflorescence, they are usually pollinated by. Many times NTA has asked this question, my dear students, in the history of NEET exam. Come on everybody, give it a try. Come on everybody, give it a try. Come on everybody, give it a try. Flowers which have single ovule in the ovary are packed into inflorescence and usually pollinated by. Come on everybody, they are usually pollinated by wind. Please remember this. This is straight from NCERT. They will be pollinated by wind. Remember this. Please remember this. They will be usually pollinated by wind. Please remember this. Is this okay everyone? Next question. Pollen tablets are available in the market for. Pollen tablets are available in the market for. Pollen tablets are available in the market for. Come on everyone. Pollen tablets are available in the market for. So they are used for supplementing. They are used as food supplements. They are known to increase the efficiency of athletes and running horses. So it is in recent market, it is becoming a fashion to obtain pollen and pollen products in form of syrups or tablets. They are considered nutritive. Yeah, next question. Double fertilization is. Double fertilization is. So which of the following matches with double fertilization? Come on everyone, what is double fertilization? What is double fertilization? Come on everyone. Just few more questions and we will be done. Come on. Double fertilization is syngamy plus triple fusion, guys. Syngamy. What is syngamy? Syngamy means when male gamete fuses with the female gamete to form a diploid structure known as zygote. इसे ही कहते हैं syngamy. Now what is triple fusion? What is triple fusion? Triple fusion means when a male gamete fuses with two polar nuclei. Two polar nuclei. Remember, just before fertilization, just before fusion, the polar nuclei fuses to form secondary nucleus, right? So whenever the male gamete fuses with two polar nuclei, each polar nuclei is going to be haploid. So there are two polar nuclei. So during triple fusion, there are three nuclei, three haploid cells that are, sorry, not three haploid cells, two cells, right? So please notice, because inside the central cell there are two nuclei. This is also one very important conceptual question that you need to understand. So during triple fusion, there is one cell which is the male gamete and another cell is the central cell which has two nuclei, two haploid nuclei in this. So this is fusion taking place between three nuclei, one of male gamete and two of the central cell, right? Hence it leads to a production of a triploid structure known as PEN, primary endosperm nucleus, which is triploid. So this triploid structure PEN is produced via triple fusion, and the central cell will now, after triple fusion, will be called as endosperm cell. It will divide repeatedly to form endosperm tissue, which will provide nutrition to the developing embryo. So this is the whole concept of double fertilization, which is a characteristic feature of angiosperms only. Please remember this, right? So double fertilization is syngamy and triple fusion. So is this question clear now, everybody? Is this question clear? Hiba, Shakir, Durga, why you guys are removing the answers? You should not accept the mistakes and don't repeat it. Make it a point. Make it a point. Be determined. Do not repeat it. That's all I need. That's all I expect you from this session. Okay? Is this question clear? Say yes or no in the chat box. Next question. Persistent nucellus is seed in seed. Listen this. There are some seeds. There are some plants, rather I should say. There are some plants In them, their seed consists of some remaining parts of nucellus. Nucellus was part of ovule, right? So there are some seeds in which nucellus get retained in the mature seeds. So can you tell me what is the term given to this reminiscent of nucellus which is found in seeds of black pepper and beet? तो black pepper और beet के जो seeds होते हैं वो perispermic होते हैं because they have perisperm which is the reminiscent of nucellus in them. So persistent nucellus or remaining part of the nucellus which is found in seeds is known as perisperm. It is seen in beet and black pepper. Excellent. Next question. Which of the following statement regarding post-fertilization development in the flowering plant is incorrect? Incorrect. So after fertilization, after syngamy and triple fusion, what are the changes that will take place in the parts? What are the changes that will take place in the flowering plant? Come on. Ovules developing into embryo sac does not take place after fertilization. Ovules actually will develop into seed. So जहाँ पर ovules थे अब वहाँ seed बन जाएंगे after fertilization. This was wrong. Ovary will develop into fruit. Ovary wall will form fruit walls. Zygote will develop into embryo. Is this clear, my lovely kids? Is this clear? Ovule के अंदर embryo sac है. Embryo sac के अंदर female gamete है. Female gamete से fuse करके कौन बनेगा? मेल गैमेट फ्यूज करके जाइगोट बनाएगा. तो जाइगोट कहाँ बन रहा है? ओव्यूल के अंदर ही बन रहा है और यहीं पे जाइगोट एम्ब्रियो में कन्वर्ट होगा, राइट? सो देन व्हाट विल सीड इज गोइंग टू हैव? सीड विल हैव कॉटलिडन्स, माय डियर स्टूडेंट्स. सीड विल एक्चुअली हैव द एम्ब्रियो, right, my dear students? An embryo is going to have the cotyledons, the embryonal axis, etc. Is this much clear, everyone? Is this question clear, everyone? Central cell develops into endosperm. I have already told you this. Next question. The coconut water from tender coconut represents. Coconut water from tender coconut represents. Coconut water from tender coconut represents. Come on, what is the nature of this coconut water? Come on, everyone. What is the nature of this coconut water? It is free nuclear endosperm. Remember, in coconut there is a part that you drink and there is a part that you eat. The part that you drink and the part that you eat both are endosperm, but the part that you drink is free nuclear endosperm and the part that you eat is the cellular endosperm, the white kernel portion. Excellent. Free nuclear endosperm, coconut water. Next question. Okay, that's the last question of this session, my lovely kids. Let me see how many of you can crack this one. Seed formation without fertilization in the following plants involves the process of. तो plant में seed तो बनेगा पर fertilization के बिना. That means without fusion between male and female gamete. So through which mechanism it is produced? Come on, everyone. Excellent. It is produced via apomixis. So whenever a seed is produced by fusion between male and female gamete, the process is known as amphimixis. This is what we do in the entire sexual reproduction in flowering plant. So entire thing is explained. The whole process is amphimixis that we do learn, understand, teach in sexual reproduction in flowering plant. And the last portion, apomixis, means production of seeds without fusion of male and female gametes. That's all, kids. We are done. So these were the most important 50 questions that you should do, you must do before sitting for your NEET 2022. On 17th of July, I am pretty sure, I assure you, this is going to elevate your botany score if you practice them again and revise the mistakes that you do. Right, my dear students? Please eliminate your mistake. Please revise the concepts where you made the mistake earlier. So till now, if you have not joined Infinity Learn NEET Telegram channel, join it right now. The PDF of this class will be available on the Telegram channel. The link is in the description. Also note, my dear students, subscribe to Infinity Learn NEET channel. On 17th of July. We will be going live just after your exam and discuss the answer key. We will discuss the question, right answer, solutions, etc. all live on Infinity Learn platform, Infinity Learn NEET channel, right? So please subscribe, hit the like button, hit the bell icon so that you do not miss out the notification, and you can also set your reminder. The class is already published, the session is already published. Set your reminder for the 17th of July after your NEET exam. Next question. So share this session with all those who are also preparing for NEET 2022. That's very important session for everyone. Hit the bell icon for the notification. Thank you everyone who attended this session. Thank you everyone for participating. That's all from this class. All the very best for your coming NEET 2022. That's all from this class, my lovely kids. On Monday, we will be meeting again on the same time, 3:30 p.m. Please note this, on Monday we will be meeting again and we will finish off the entire botany. So one more session. This was session number 3. So the last session, that means the session 4th, will be on Monday, where I will discuss with you the remaining 50 questions from your molecular genetics, Mendelian genetics, plant breeding, microbes and the entire ecology unit. So this for the important 50 questions related to these chapters and this unit we will be discussing on Monday 3:30. Do not miss out the class. Set your reminder, join it and let's solve, let's revise, let's try to understand the important concepts on which NT has already asked question from those chapters. So that is going to be the final session from this Botany 50 series questions, my dear students. Please do not miss out that class. Thank you very much for listening. Thank you very much for joining. That's all from this class, my lovely kids. Bye-bye. Take care. Study nicely.